Hello, good afternoon, everyone. Thank you for coming to today's seminar. This week, the seminar is being hosted by the Herbarium. It's one of our annual Wagner lectures. And this year, we have the pleasure to welcome Dr. Andrew Hipp. He is the director of the Herbarium at the Morton Arboretum in Chicago, where he's also a senior scientist in plant systematics. Andrew is a specialist in oaks, sedges, in plant evolution and speciation in general. And he has a cool connection with us through two of our emeriti curators, Tony and Paul, who did previous work with him. So that's really cool too. And yeah, so without further ado, let's welcome Andrew.
All right, thank you very much. Can you hear me okay? Yeah. All right, thank you very much for the introduction and for the generous invitation to speak here today. It's really an honor to be back here. It's been, I think, a decade since I've been here to work with Tony and Paul in the herbarium, and nice to be back here today to talk with you about some of our current work in bur oak and its relatives. I must confess, I've got enough. I don't know if I have 30 minutes of slides here or 2 hours worth, but I promise I'll cut it off at an hour. So oaks. Oaks arose at or before the Paleocene, Eocene thermal maximum, about 56 million years ago. It's a fairly old genus worldwide, about 425 species. Within about the first 10 million years of oak evolution, all the major lineages that we know, or most of the major lineages that we recognize today, had arisen already. So we could have recognized red oaks, and we would have recognized white oaks, intermediate oaks, ring-cupped oaks. We wouldn't have recognized the particular species, but we would have known the lineages that we see today. So when you go out on the landscape and you look at oaks, let's see if this is going to work. That I don't think is going to work. That's okay. But this will, won't it? Maybe this won't, but this will, won't it? No, I'm not going to have any pointers. So I'll point with my fingers. As you look at white oaks, that section Quercus, or red oaks, that section Lobati, these are all different lineages of oaks that we recognize today. The white oaks are what we're going to be focusing on today. This is a section or a clade that arose most likely in northern North America around 45 or 50 million years ago. Bur oak.
Oh, there you go. I think this will work.
Thank you very much. Thank you, Thais. Bur oak, eastern white oak, California valley oak, and about 145 other species, including 25 in Eurasia. These are all white oaks. They all have annual acorns, which means that they're pollinated in the same year that the acorns are produced, which is not true of most of our red oaks. Their nut walls are smooth on the inside, not hairy. None of them have bristle-tipped leaves. They have very distinctive whitish bark that you can recognize from Canada all the way down to Chiapas. It's a really distinctive clade. And white oaks have been a real global success story. This is the shaded areas are the range of oaks worldwide, and this hashing that goes from northwest to southeast, that represents the white oak clade. Anywhere in the world where you go and find oaks, for the most part, you will find white oaks. They've covered the globe, with the exception of the area where you only find ring-cupped oaks down here, and one little area where there's just a single red oak in Colombia. As we zoom in on the white oaks, there's an so this is the clade that just includes the white oaks. Within the white oaks is an eastern North American clade right here, actually a grade, comprising mostly species that straddle the southern margin of the last continental glaciation. And bur oak, this species right here, is part of this clade. So bur oak, if you look at the genetic variation in bur oak, what you're looking at here is an ordination of single nucleotide polymorphisms a small number of them, 75 sampled from across the genome, and we've just ordinated them. We've put them into 2 dimensions, taken all the pairwise genetic distances, and said, for each individual tree, who's most similar to you genetically? And whoever's most similar to you genetically falls close to you in ordination space, all thing, for the most part. And what you find is that bur oak, Quercus macrocarpa, is distinct genomically from all the other species of Eastern North America. It does exchange genes with them to a certain extent. That's a topic we'll talk about more in tomorrow's lecture at the Herbarium. So it is in this way a very typical oak species, ecologically, genomically, and morphologically distinct. But it has a remarkably broad geographic, morphological, and ecological range, making it rather exceptional, an exceptional species in an already exceptional clade. So if you look at its range, here's Quercus macrocarpa. It ranges from Manitoba down to the Gulf of Mexico, from the Black Hills out to New York and Vermont. A comparable range to eastern white oak, but morphologically much more diverse. We think of it as a savanna species, so very often we find it in these park-like conditions. But bur oak ranges to dry cliff tops, to forests, to towering trees in bottomlands. Bur oak is named for the morphology of the species of the acorn cap. The acorn caps form these burrs at the margins. They're very diverse in size. They can range from about dime size at the north edge of their range to large enough to fill the palm of your hand in Texas and Oklahoma. Leaves have this very distinctive look, sort of a wasp-like constriction at the middle, and they're hairy on the undersides. So with this combination of characters, you can recognize bur oak no matter where you go, whether you're in Canada or in Texas or in the Black Hills or exotic Peoria. And even when you see the teeny tiny leaves, these little bitty baby leaves, distinctive even at this stage where you can sort of see their ribs. So this is bur oak. All a preamble to tell you about this cool study that I've been involved with collaborators from across North America and in China. This is a Dimensions of Biodiversity project funded by National Science Foundation and the National Science Foundation of China. 6 institutions in the US and 4 in China working together to understand how gene flow between white oak species, with bur oak as one of our focal species, influences the communities of symbionts that live on trees. As you can imagine, these are the PIs who originally submitted the proposal, but the lion's share of the work is done by postdocs, by graduate students, by undergraduates, by research assistants who've worked with us, and I want to acknowledge them here as well. The focus of this project is to understand how phylogeny, evolutionary history, genetic diversity within species, introgression, the movement of genes between species by hybridization, and functional traits interact to shape the communities of organisms that live on oaks, so the fungi and the insects that live on oaks. It integrates 3 projects overall that address genetic, phylogenetic, and functional dimensions of biodiversity on both bur oak, Quercus macrocarpa, the one we'll talk about today, and Quercus serrata, an East Asian oak. All the projects I'm going to be talking about today I'm talking about the North American side. These are all run in parallel in China on Quercus serrata. That'll be topics of future talks. Today I'll focus on bur oak and 2 areas of our work. Variation in common gardens and diversity and composition of bur oak symbiont communities, mostly sampled from across the wide range of bur oak. If you're interested in gene flow, in hybridization, in what makes a species, in how gene flow can affect species, that'll be the subject of tomorrow's talk in the herbarium. So be there or be square. Okay, so first, variation in common gardens. Let's go back to Darwin, who in 1859 gave us a mechanism by which evolution operates. This is natural selection. So evolution by natural selection is inevitable when there's variation in organisms, when that variation is heritable and affects fitness, and when resources are limited, right? So evolution all starts with variation. And natural variation has two main causes. Or two main sources. There's plasticity. This is variation within a single genotype. If you take a tree and you clone it and you plant it into 6 different environments, it's going to grow taller or shorter, have different shaped leaves, produce more or less fruits, just based on the resources that are available at the site. That source of variation is mostly not heritable, aside from some epigenetic effects. Genetic variation, on the flip side, is heritable. This is the variation that is passed down through genes that makes siblings look similar aside from their shared environmental conditions and shapes regional differences. Teasing apart these two sources of variation is essential, because if you want to understand how trees are going to evolve in response to climate change, you got to know what of the variation is actually heritable, because that's the only part that evolution is going to be working on. As Darwin was grappling with variation and natural selection, and he's writing on the origin of species, foresters who were growing trees were learning to tease apart plastic and genetic variation in trees for a very practical purpose. They wanted to know what trees would grow best in their areas. So the first provenance study, a study of trees from different provenances, different sources, was a Scots pine planting at Les Bars, planted between 1823 and 1850. And this planting told them that Scots pines from different areas perform differently, giving them insights, giving the foresters at that time insights as to which Scots pines they should be planting. Oaks weren't far behind. The first provenance tests for Quercus robur and Quercus petrea, Quercus robur, which is called the French oak if you're in France and the English oak if you're in England, were established in 1877. These provenance tests can be used today to investigate how oaks evolve in response to climatic gradients. So provenance trials were the first common garden experiments. A common garden experiment is an experiment where you take multiple genotypes from different areas or multiple species from different geographic regions, and you plant them together into a common environment. What that does is allow you to look at the variation within that garden with, if you have enough replication, gives you an estimate of how much genetic variation, how much of the variation you observe is due to genetics. Variation within a common garden, if it's done well, reflects genetic variation. Variation between genotypes among gardens reflects plastic variation. Common gardens, by planting these multiple populations together, show us, for instance, that in oaks, what we look at here is the transfer distance, sort of the difference between the source population and the population where it's planted in a common garden, in the difference between annual precipitation and evaporation. And that difference in dryness predicts how well the oaks perform. And what you see is that on average, oaks are locally adapted. They do best in the environment where they were best planted, where they arose, where they evolved. And there's huge variation. The variation along the y-axis shows you how much genetic variation there is within populations. So common gardens, provenance trials were a jumping off point for Goethe Turasin, who in 1922 coined the term ecotypes based on his work with common gardens, where he planted Rumex acetosella, sheep's sorrel, collected from wind-swept shorelines versus more protected inlands, and he grew these different genotypes of a single species together in a common garden and asked this simple question: do they maintain their differences, or are the plants at the shoreline just beaten down by the environments and showing a plastic response? And in fact, they showed a genetic response year after year after year. The shoreline plants continued to look short and stumpy compared to the inland plants grown in the same site. That's the difference. That is a genetic difference. This work is probably more familiar to you from Clausen-Keck and Heisey and Hall, ignored by Clausen-Keck and Heisey in all their publications, whose famous experiments in California demonstrated that species differences are genetic. They collected initially hundreds of species and settled in on a couple of dozen that they planted along transects from Stanford down at sea level through Mather up to Timberline to ask how different populations of, in this case, yarrow, Achillea lanulosa, would perform in different environments and to what degree these differences are genetic. Hall, Clausen, Keck, and Heisey transplanted plants that they collected in multiple environments back into that full set of environments. This is called a reciprocal transplant experiment. It's the establishment of multiple common gardens near the sites of plants that are collected or organisms that are collected from these different environments. By transplanting them reciprocally in both directions, you can see whether or not plants perform better or more poorly in one environment over the other. If they're locally adapted, if they do best where they've evolved, you'll expect to see that they perform best in that environment. So by doing these transplants, Howell, Clausen, Keck, and Heisey showed that the gene combinations you inherit matter. The highest elevation populations suffer the least in high elevation habitats. So our past work in wild populations of bur oak had shown that variation in bur oak traits, in this case lobedness, varied from north to south. Here we see a gradient in circularity, which counterintuitively was calculated such that the more lobed trees have the higher circularity. The less lobed trees have lower circularity. And what you see is that the highest lobedness and the highest circularity is among the southernmost plants. But in the wild, we can't distinguish whether or not this variation is just plastic, just the environment acting on a given genotype, or whether that's genetic. The only way to tell that is to do a common garden. So what I'm going to talk about now is this reciprocal transplant experiment, these set of common gardens that we're undertaking in bur oaks at 3 sites that span a latitudinal gradients from Cedar Creek Ecosystem Science Reserve at University of Minnesota to the Morton Arboretum, where I'm based, down to Oklahoma at the Kessler Atmospheric and Ecological Field Station. The goal here is to understand how much of the prodigious bur oak variation we observe is genetic and how that variation affects tree fitness or performance. So the design here, which was set up by Janine Cavender-Barris, is a sort of a standard quantitative genetic design. Our expectation is that populations would be locally adapted, that the Oklahoma garden of trees would perform best in Oklahoma, that the Illinois plants would perform best in Illinois, that the Minnesota plants would perform best in Minnesota. So to effect this project, we collected acorns from natural populations in 2018 and 2019 in the vicinity of the experiment. So these are the 3 sites. Each star shows you the location of the common garden, the reciprocal transplant replicate that we planted. In Minnesota, you can get a sense of where the trees were collected. In Illinois, the acorns rather, you can get a sense of where the acorns were collected. And in Oklahoma, you can get a sense of where the acorns were collected. Each of these sets of acorns was then sent to Indiana, where they were grown into a sandy bed, basically, where they could get used to the outdoor environment, growing up in a common environment, for transplanting back into the 3 common gardens. Here's what it looks like in Valonia. These were planted by mother tree. We collected multiple seeds per mother tree, planted them out into these sand beds separated by mother. And then these were basically dug up by Bob Hawkins and his crew who grew the trees, and shipped to each of our 3 sites for planting. While these were being grown, we prepped the site. Each of our sites was treated really violently. They were tilled, they were herbicided, they were planted with a cover crop, they were tilled again, they were herbicided again. Enough stuff that it's hard to sleep at night. And then for each of the trees, we were planting 600 in 2021. We augured a hole for each of our bare root trees that were coming in. And then we drove down to Valonia to pick up these bundles of trees. Here's a bundle of trees from the acorns from one mother tree. this acorn lot was set, we had one set that was sent to Illinois, one to Oklahoma, one to Minnesota. So we've got replicates of each mother tree planted out into each of the common gardens. They were laid out in a randomized block design, planted out over the course of a couple of weeks. In some cases, the roots were a mess, so we had to spade them in, watered them in, tagged them all so that we knew what source population, what Minnesota, Illinois, or Oklahoma, what source site within that source population, what mother tree they came from. Weeded, collected some preliminary data, mulched the living daylights out of them, and cared for them over the course of 2 years to get them established. And this is what the Morton Arboretum experiment looked like in 2021. You can see the distinction already at the end of that season between the deeply divided Oklahoma leaves and the less deeply divided Illinois leaves. In 2023, with already acorns being produced, we'll come back to that. That was a shocker. I know you're shocked. And 2024, here with our colleagues from China visiting. Okay, so that's setting up the experiment. What have we found so far? You can see that there was a great deal of excitement as we began to gather data. So first findings, a basic question, which is going to seem like I'm setting the bar really low. Did we actually plant what we meant to? And any of you who've worked with oaks know that oaks are a little bit promiscuous. They do hybridize. So we genotyped all the trees that we had planted and genotyped along with them individuals of the species that they might have hybridized with to make sure that we weren't planting a lot of hybrid acorns. We got low coverage genome skimming data, about 186,000 SNPs from across the genome, enough to validate species identity. You'll notice that part of chromosome 1 seems to be missing from our SNP set. That was an artifact of the code that we used to generate this picture. So what you're looking at here, if you haven't seen one of these, this is an analysis that just asks of each population, of each individual, which is a horizontal bar here, what percentage of its genome harkens from, in this case, each of 6 ancestral populations inferred. And we threw in a bunch of species, eastern white oak, swamp white oak, lyre-leaved or overcup oak, a bunch of our Quercus macrocarpa from the garden, a swamp, a chestnut oak, chinkapin oak, post oak, and asked, are any of these hybridizing to produce the acorns that we find? So the first thing we found is that there was a north-south split between the oaks that were sourced in Illinois and Minnesota versus those in Oklahoma. That's a split that was already known from our previous work. I'll discuss this in some gory detail tomorrow. When we aggregate all the mess that is here in bur oak, just to focus on introgression. So here what we're going to do is aggregate the multiple populations that were discerned to be in bur oak so that we can see the introgression effect from other species. What we find is this trickle of introgression, not much, probably a lot of it is not even significant. What's striking here, though, is everybody's always like, oaks are so promiscuous. You go out there and collect seed, it's all going to be hybrids. We've got, this is just, I think 300 of the experimental seeds successfully genotyped for this initial trial. Not a single one of them is an F1 hybrid, right? So we did succeed in actually getting bur oaks, but we do see a trickle of gene flow in the mother trees with a background level of introgression from eastern white oak, predominantly in the northern trees, and chinkapin oak, predominantly in the southern trees. Again, more of the range-wide genome data tomorrow in the herbarium. It's going to be fun. Second finding. So that was great. That was satisfying. And nice to know that these trees are not just completely promiscuous. Second finding was that trees flowered a lot earlier than we expected. This is a paper by my postdoc, Rebecca Mohn, who's also analyzing the genomic data. As a naturalist, as a botanist, I had always figured 20, 30 years to get an oak to go to acorn. And when you look in the seed production manuals, the woody plant seed manual here. They say, yep, the minimum seed bearing age for Quercus macrocarpa, like 35 years. But we found acorns being produced in 2023, which is 4 to 5 years after our first acorn, and predominantly on trees that were larger. So here we see July 2023 acorns on 6 trees, 4 to 5 years after collecting. And in fact, they weren't inviable acorns, they were viable. They grew into little seedlings here with Rebecca's help. Interestingly enough, those first trees, we generally expect to see male flowers earlier on plants because male flowers usually take less work to produce than the fruits. They're lower energy. We saw no male flowers at all for the first couple of years. Instead, what we found was pistillate flowers, female flowers, and acorns only. So when we look at the flowering data from 2023, 6 trees flowering, all pistillate, no boys. 2024, 16 trees flowering, all pistillate, no boys. And 2025 flowering, we had flowering on quite a number of the trees. Only 2% of them were staminate only. The rest were pistillate and staminate or pistillate only. Why should this be? One hypothesis Rebecca's raised is that it actually may be advantageous for a small tree to produce pistillate flowers only if it's wind pollinated. Reason being that if you waste your energy producing pollen and you're a wind pollinated tree, if you're a tiny little tree, that pollen's not going to go anywhere. It's going to get out into the forest, fall on the forest floor, crash into trilliums, do no good. If you're a big tree and you're shedding pollen, a small tree can still capture that pollen and fruits. So there may be selection for early production of pistillate flowers. Just a hypothesis. Third finding, all the populations do best in Illinois. So that's plastic variation. And it's probably because Illinois is not as cold as the Minnesota site where everything was frozen back to the ground every year. It's not as dry as Oklahoma where all the Minnesota trees just would die off in, not all of them, but like 50% of them. But growth and survival nonetheless have a strong genetic component. So within each garden, so here's each garden, right? You see variation where the Oklahoma trees seem to be doing best at each site. Variation in growth and survival is predicted by physiological traits. That's very satisfying. And those physiological traits are themselves heritable, right? They have a strong genetic component and enough genetic variation within the population that natural selection can act on them. And that's particularly true in the southern populations. This is work by Lucy Ray, who's a postdoc in Janine Cavender-Barris's lab. Okay, that's important because that's the variation that natural selection can act on. Similarly, genetic variation for leaf lobedness, how lobed a tree is, tracks climate in the same way as field measurements and matching expectations. In other words, we see more lobed leaves among the Oklahoma trees than we find among the Minnesota trees. Lobedness also shows more divergence among populations than we expect based on genotyping neutral markers. This is a test for natural selection. This is to say, if you look at the variation in lobedness and look at the difference between populations and compare that with the variation in a bunch of neutral genetic markers, in this case, those SNPs that are scattered across the genome, and their differences among populations, the differences in lobedness between populations exceeds the difference between populations in those neutral markers. That's exactly what you expect if natural selection is going on, because it's pulling those, the lobedness, causing those populations to diverge from one another in lobedness. When you put these things together, the variation in traits, that's the heritability, that translates to heritability, and the divergence between populations due to natural selection, traits are expected to evolve in response to changing climates. And this is the vector of expected change in leaf mass area, which is the mass of a leaf relative to its area, which is a good predictor of response to water availability. The change in relative growth rate. These are going to evolve in response to climate change. What we don't know, however, is if they'll evolve fast enough. So this is the first main project of the grant. It gives us a good idea that this variation that we see in bur oaks is functional and heritable. Now the question is, how does this variation affect the symbiont communities, the communities of organisms that live on and depend on bur oaks? We know that oaks are disproportionately important ecologically, and a lot of this has to do with their mass. This is a big chinkapin oak outside of Lexington. It's called the Great God Tree by its owners. They own a horse farm. They call it that because they look at it and they're like, great God, that's a big tree. These oaks are massive organisms, like these 330-year-old Quercus petrea that were planted under the auspices of Jean-Baptiste Colbert under Louis XIV, who in the mid-1600s, in the heart of the Little Ice Age, planted these Quercus petrea, figuring, okay, we're going to need big white oaks to make our Navy, like in the year 1900 and 2000. By the time these trees were big enough to make ships, what France really needed was wine barrels. And so those trees are now being used for wine barrels. It's the sheer mass of them that gives them their impact. It's the mass that allows us to make charcoal, like this pollarded tree in Mexico, where they're basically cooking it to make charcoal. So the mass of material, the biomass of oaks, which exceeds the biomass of any other woody plant genus in North America and in Mexico, is part of its ecological importance. But the other part is its sheer diversity. The number of species that we see in oaks, which is higher in the US and in Mexico than any other woody plant genus, is essential to their effect on ecosystems. Because the more species there are, the more ways there are of shaping ecosystems. As a consequence of diversity, differences among oaks influence endophytic fungi in the leaves. So this is some of the preliminary data that we generated for the grant. Each of these dots is a sampling location of one tree that we sampled from on the landscape for endophytes, the fungi within the leaves. And you can see what species we sampled. And when we sequenced the endophytes within the leaves, each of these points then is one tree that we sequenced. And the position on this ordination reflects the composition of the endophyte community within the leaves. And what you see is that bur oaks and eastern white oaks and swamp white oaks are differentiated to each other to a certain degree in their endophyte communities. And when we look at the gall wasps, whose young grow up inside of oaks, we see a similar difference. Each of these dots is a gall wasp community on one tree of Quercus douglasii versus Quercus lobata sampled in California. And what you see is that those gall wasp communities are also very different. So these species level differences matter, but at the start of this project, we had no idea what the gall wasp, the endophyte, the mycorrhizal communities were like on bur oak and its close relatives. So let's look at the gall wasps first, which is work that is done by my colleague Ian Pierce, who's also done wonderful work on masting in oaks, if you're interested in that. So just if you haven't seen gall wasp, oak galls before, this is a typical oak gall on Quercus sapotifolia in Oaxaca. This giant thing is tissue that is created by the oak tree. When a gall wasp comes and puts her ovipositor on the tree, lays an egg, she basically hijacks the tree's genes and changes gene expression so that the tree builds a home for the egg that she lays inside there. So she's laid an egg right in the middle. This is the same gall split open. And there you can see that that egg has hatched open, and there's the gall wasp larva inside doing what it's doing, which is resting at this point. So Ian and his crew covered this range of this portion of the bur oak range in the fall of 2022 and 2023, sampling galls. So they sampled using binoculars to observe gall morphology, using pole pruners to cut samples down. They identified them in the field. This is Gabe Rybakov, our undergraduate, who actually wrote the paper that I'll be talking about tomorrow at the Arboretum, underneath the great god tree. And they sampled galls like Quercus, Androcus Quercus frondosus, Neuroteris, Dishels Caspis Quercus mama, the Androcus dimorphus, Acraspis, and like 30 other species of galls, each more fantastic than the next. And then they took these back, and Leah Samuels, who worked as a research assistant on this project, took them all back to the Arboretum and reared them out. Grew them in these little salsa containers to see what was growing inside each gall. Because we had, for many of these, no confirmation of what species was inside them. And so, for instance, here's what she found popping out of this bullet gall right here is a Dishel Caspis Quercus mama, emerged from a gall collected along the Hudson Rail Trail in Vermont. And also some parasitoids, some that are parasitizing the gall wasps inside the galls, some that are just taking advantage of the gall, living adjacent to the gall wasp. So here's a Taurimus that came from one of those same galls that she reared out. So lots of fascinating stuff that's come out of this just in terms of what species are living on what oaks. What have they found so far? Well, one thing is that oak species, what species you are, affects gall richness and gall abundance. So here's the oak species that they sampled from. And what you see is that the abundance and the richness varies among species. So some species are particularly good for galls, some species are particularly poor. More interestingly, though, when you actually look at the gall wasp communities, here's a clustering diagram of gall community similarity, where the proximity of two individuals on this tree just reflects the similarity of the gall wasp communities. That fairly closely tracks oak evolutionary history. This tells us that the similarity of two oaks in evolutionary space influences the similarity of their gall wasp communities. Okay, so that's the galls. Oaks also host really diverse mycorrhizal fungi. So to get at the mycorrhizal communities, roots were sampled across 30 sites in eastern North America. Bur oak was sampled most, followed by eastern white oak and all the co-occurring white oak species. This is work that's being analyzed by Kathleen Thompson, and the sampling effort was led by Peter Kennedy and his lab before Kathleen started on the project. Now, they were looking at two different kinds of mycorrhizal fungi. One is ectomycorrhizal fungi, which live on the outsides of roots. One is arbuscular mycorrhizae. These are fungi that penetrate the roots. Both types occur on oaks. Some trees are dually colonized. Other trees are colonized by just one type or the other. So they wanted to understand for both the arbuscular mycorrhizal fungi and the endomycorrhizal fungi whether abiotic factors, the degree to which environment and soil and climate were affecting the mycorrhizal community, and biotic factors, what oak they were growing on, and what the neighboring forest community was like. So they sampled roots and soils from 30 sites across the bur oak range in 2022. At each site, they took a soil core as well as a root sample that they could take back and sequence to look at what fungi were growing within it. They calculated fungal presence at each site, found a lot more ectomycorrhizal fungi than arbuscular mycorrhizal fungi, things that you would recognize on a walk through the woods, things like brittle gills, things like stinking earth fan, jellied false coral fungus, webcap, amanita, chanterelles, and some things that you would not see at all, like truffles, which are fairly abundant in their data set. Okay, so when they analyzed these data, what did they find about how oaks are affecting these symbiont communities? The first thing they found is that oaks have arbuscular mycorrhizal fungi in their roots, which was important to document. It was not clearly documented how frequent that was, but their endomycorrhizal fungal communities were far more diverse. This gives you an estimate of what their richness is of the ectomycorrhizal fungal communities versus the arbuscular mycorrhizal fungal communities at each site sampled. Second thing that's important is the forest community around each host tree strongly shapes the diversity and composition of the mycorrhizal fungal community. This is from their range-wide, the ectomycorrhizal fungal community, not for the arbuscular mycorrhizal fungal community. This is from their range-wide data. We also have data from an ecosystem, a biodiversity ecosystem function experiment where we're controlling diversity. They found the same thing. This is pretty preliminary data, but it supports the same basic finding. And the third thing, importantly, is that the ectomycorrhizal fungal communities vary depending on the host oak species and to a certain extent by hybridization. They have less data on the hybridization, but what you see is that each host species is differentiated, if weakly, in its mycorrhizal fungal community from the others. Interestingly enough, we've looked at the genetic data for the families, the half-sib families that we planted into the bur oak common garden, and we find a similar pattern of differentiation at the genetic markers that we sampled. Whether or not symbiont communities are variable among families in the same way remains to be seen. And that's the next thing that Kathleen will be looking at, because this past October, she and a whole group of people spent a week out in the field with us digging up roots around each of the planted trees in our common garden to ask this question: are the mycorrhizal communities that we see assembling on Minnesota versus Illinois versus Oklahoma trees, are they differentiated, right? Are the genotypes of the trees filtering differently from the mycorrhizal communities that are already present at our site? I have to say, it was a thrilling week of digging up these roots, and you trace these roots away from the tree to get just a little bit there, and you put that in the bag. It was like living beneath ground for a week. You got to try it sometime, my friends. Okay, so where are we now in our understanding with respect to these questions? So far, the common garden shows us that genetic diversity of trees shapes their functional diversity. All right, so we've got that part nailed. Range-wide symbiont data and the biodiversity ecosystem function data show us that phylogenetic diversity, both the phylogenetic diversity of the forest around a host tree and the phylogenetic identity of the host tree, shapes symbiont diversity. And genomic data shows us the magnitude of integration. Again, we'll talk about this a lot more tomorrow. In the coming year, here's what we'd like to better understand, is how genetic diversity and integration are both shaping symbiont diversity. And our goal ultimately is to develop this composite picture which integrates the evolution of species, ranges on the landscape, the assembly of communities in which species integrate and compete, and the effects of evolution of tree biodiversity on both of these. Because you really can't understand the effect of any organism on the forest or the forest on an organism without understanding all of these pieces. And ultimately, my goal is that even in these troubled times, that this kind of research drives people back out to the forest to get an appreciation of the trees in situ. These are fantastic organisms, widespread, beautiful, and my hope is that that this work just gives people a little bit more joy in seeing them. This work would be impossible without a ton of collaboration. Most of what I presented today is by our collaborators. Tomorrow we'll present on more of our own work. It wouldn't be possible without support, particularly collaborative support between the National Science Foundation in the US and China. My lab group, without whom this work would be a misery, or at least not as joyful as it is. And my old advisor, Paul Berry, here shown in UW-Madison, pollinating Amorphophallus. Paul took me on as a graduate student when I didn't know anything. He was very charitable and kind and taught me much about leading a lab as one sort of helps one's family along. And Tony Resnicek, who has been a support, a leader in the field, and a great advisor, co-advisor to me as well when I was working on sedges, and continues to be. And thank you to all of you. I encourage you to be in touch if you have questions, and I appreciate your time. You could have been anywhere today, and you chose to be here. So thank you very much, and I'll take questions. Yes. I think you're supposed to use, is this thing?
Oh.
Is this thing? Or yeah, I don't know how this works. Okay.
One of these works. We'll use that to head this way.
I don't know how you turn it on.
Really? Hello, hello. There we
go. First of all, that was a really cool talk. More like, sorry, it's my first mic. First of all, really cool talk. Leaf shape variation in your common garden experiment. I was wondering if you had any insight into like what you think might be like the driving forces behind all that variation you see, and also the kind of like decline you see between the north and the south.
You bet. Yeah. The great question. So the observation was that in the common garden there's a lot of leaf shape variation just within Quercus macrocarpa, bur oak, and you observe that in the landscape as well. And there's co-grading, they're varying in the same direction, mercifully. As you go north, you tend to see smaller leaves with less lobing. As you go south, the growing season gets longer, so the leaves have more opportunity to grow. We think that's one thing, but they're often in drier conditions. So lobedness does a few things. It allows the leaf to cool down, and it reduces overall area while the tree grows larger. So it's almost like producing more smaller leaves. So we think it's a response to drought stress. Yeah.
About the distribution of Quercus
Okay.
in the world.
Yes.
And it's on 3 continents with disjunct distributions today. Were they, but they originated a long time ago. What accounts for that disjunct distribution? Is it dispersal, or is it because the continents were, the eastern and western sides of Eurasia won't be a continental, a tectonic problem. But what about that?
No, that's a great question. So the question was, what accounts for this wide distribution that we see in oaks today? The ancestor of oaks was evolving at 56 million years ago or so, at a time when there was free transit between North America and Europe. Mammals could walk across, but could move between the continents till about 49 million years ago. And there was movement of plants even later than that. But from about 50 million years on, temperatures start dropping after the early Eocene, thermal maximum there, and they start. So oaks get pushed downward. North America at that point was covered with broad-leaved evergreen forest that is shoved southward and sort of, extirpated against the coast or driven down into Mexico, and oaks just start marching inexorably down with the other temperate flora. Same thing is happening in Europe. Now, white oaks do a trickier thing. White oaks, so that's the sort of oaking of the continents initially. At about 20 million years ago, oaks, the white and the red oaks both moved down into Mexico, and they just go bananas. There's like 180 species down in Mexico now. That's a really cool story on its own. But the white oaks in Eurasia get back from eastern North America. They appear to be sister to 3 species in eastern North America: Quercus alba, Montana michoii. And they go back either by the North Atlantic land bridge, which was traversable until about 10 million years ago. It looks like intermittently. Or the Bering land bridge. We don't know for sure which is the case. They go back, and that one ancestor, that one population, gives rise to what's now 25 populations, all of whom have evidence of introgression, gene flow, from one Eurasian species, fairly distantly related, Quercus pontica, which is now only in the Caucasus. But there's evidence of introgression between pontica and the ancestor of that whole clade in Eurasia. Whether that was helped in their sort of covering of Eurasia, we don't know. Does that answer your question? Yeah.
Other questions? This one works. Yeah.
Awesome work. So as a member of the Weber Lab, I feel compelled to ask about our favorite oak symbiont, which would be mites.
Okay.
And so first of all, I'm just curious whether this project writ large has any plans to look at mites. And
I guess we do now.
and if not, I guess I'm wondering whether you see any morphological variation that might correspond to differences in the habitat the mite would have on the leaves, because you mentioned that the undersides of the leaves are fuzzy.
Yeah, great question, and nice pun on mite. Then the undersides of the leaves, so mites like to inhabit these domatia, these little patches of hairs on the undersurface of the leaves. You do see variation in the trichome, the hair density on the undersurface of the leaves. You see variation in the types of hairs, and there's high variation from Quercus swamp white oak, swamp chestnut oak, and bur oak all can have pretty densely pubescent undersides. The rest not as much, just in this one little lineage. So yeah, there's potential for mite action there, and if you'd like part of it, let's talk. Because I know Ian Pierce. Ian came to if those of you who've not met Ian is a joy, and he did come and survey our oak collection one year for mites, for domatia, and was interested in whether or not, had some ideas. So we should talk. Thanks, great question.
Others? All right. Well, Andrew, thanks very much for a great talk.
Thank you guys very much.